Now, I've mentioned before, and we're going to focus this lecture and the next couple of lectures on this thing referred to as complement. Complement is a series of proteins that are used for their antimicrobial properties within all mammals, as well as there's a version in many other animals. They are numbered C, as you see here, C1, 2, 3, 4 on through. The number designation does not correspond to activation step. They were numbered based on when they were discovered. Okay, so C1 first discovered, C2 second, so on and so forth. You will notice here in this figure 211 that the different classes of complement, whether it's activating, the binding, mediators, whatnot, C1Q, C1RS, C2A, C5B, C5A, so forth. This is to denote that for some of these, the A, the B means that it was one propeptide that was cleaved, generally self-cleaved. A has a function, B has a different function. So keep that in mind. They are the proto, pre, however you want to think about it. Protein is referred to as a zymogen. The zymogen is a protein that has secondary, tertiary structure, but is going to need to be cleaved, usually proteolytically cleaved by self or another complement protein. And then the two components will finally get their quaternary structure, which will both have some activity. It has been recognized that there are 3 pathways for the activation of this, what they refer to as a cascade. Once it starts, it's, unless there's some outside force, it will continue until you end up with this barrel embedded in some membrane. The difference between the 3 pathways starts at how they begin. Notice here, the 3 beginnings come to one middle ground, and then the middle ground gives to 3 outcomes. All 3 outcomes happen. So from this step down, which is the C3 activation, so from C3 on to the cleavage of C5, 3B acting as complement receptor, complement activator, as well as this barrel. This will happen no matter which of the 3 ways gets initiated. The different initiation are known as either the lectin pathway, the classical pathway, and the alternative pathway. So the initiation. The classical pathway is what we're going to focus on first. For classical pathway, it's classical in and only that it was the first found. First one thoroughly studied to a point, and that it was initially thought this was the one and only way. Well, then somebody figured out that, well, there are other serum proteins that like to interact with C3, or at least the C3 zymogen, and that became the alternative pathway. So the classical, this is what we thought was the big way, the one way. Later on, oh, here's an alternate. And then many years later, somebody said, oh no, there are some other pathway. There's another pathway that also will result in the activation of C3. This is referred to as the lectin pathway. Well, we kept the names classical because it was the first, alternate because, hey, look, there might be this slightly other way, and then there was a third way that we figured out. Now, when we step back and we look at the genes and the proteins that are involved, we find a few things. First, the lectin pathway we find not only in all mammals, but we find in many other animals, non-mammalian animals. And we think this may have been the oldest, one of the first ways to activate the original cascade. Now, we don't think it was always the complement cascade that we see now, but something that was very homologous to what we have now. And excuse me, lectin pathway would have been the way to activate this. The alternative pathway, you see here factor D, factor B, and the cleavage of C3. Well, we think this is a feedback loop. Once it starts, it self-regulates, self-perpetuates. So the trick is, once it starts, how do you stop it? And, for us and our bodies, we don't want this going. As we're going to see later in this chapter, how do we inhibit it from getting started? Doesn't matter which of the three ways you look at or is being utilized, C3 is where they all converge. When C3 gets activated and the cascade truly gets going, no matter which way you're looking at, you're going to look at a recruitment, as you see here, of cellular response, whether it's the innate or the adaptive cells. Here they're showing a macrophage as it's crawling out of the bloodstream into the surrounding tissues. They refer to this as diapedesis. The macrophages are following a trail of breadcrumbs. The trail of breadcrumbs being C3a that is floating away. C3, the zymogen C3 gets cleaved into C3a, C3b. C3b will adhere to the membrane of the target cell. C3a floats away and interacts with the epithelial cells lining the nearby capillary bed, couldn't think of the word, which will tell them to start interacting with macrophages and pull them in. C3b, as I said, will deposit on the surface, will begin interacting with receptors on the surface of the cell. If it's our cells, if it's bacterial, there's no receptors, and it'll start recruiting in the rest of the cascade, C5, 6, 7, 8, 9, which will form this nice little barrel pore, which will allow everything from inside the bacteria to leak out. When you look at C3 itself, C3 is a self-limiting protein. We don't want this just automatically cleaving left and right. We want this to cleave only when necessary. Now, yes, we do have some mechanisms on our cells to stop it, but we do not want those just being activated in any shape or form. You see that C3, when it's the zymogen, has the alpha chain and a beta chain held together by a disulfide bond. A convertase, known as C3 convertase, will come in and it will cut right there, releasing the beta chain and the alpha chain, C3a, C3b. C3b will remain bound to the surface of a pathogen. C3a, once it's cleaved, will be able to fold itself around, become activated, and then will recruit in more cells that are going to react to activated C3b. you get certain proteins, certain moieties that will bind to those. Bind to things that are you and things that are not you. Phycolins. It's a fibrogen-like domain that is going to bind to the different sugar moieties, those glucose sugar things, and they're going to do so in such a way that it allows for the beginning of the complement cascade. You see here the mannose binding lectins and the phycolins. The mannose binding lectins, the phycolins, if you close your eyes and squint real hard, do sort of resemble each other. A trimer of monomers that are held together by the collagen. These trimers have an end that is for sugar or fibronectin interaction. Fibronectin is going to be proteins embedded in the plasma membrane. Here we have the carbohydrates, the sugars sticking out of the plasma membrane. Now, one triplet does nothing. But when that one triplet is grouped together with 5 others, and you get this hexamer, with all of their collagen helices binding together, you now have a stable platform that will allow for other things to interact. So once these 6 trimers are anchored, whether you're talking MBL or phycolins, and they form this stable base, you will see the recruitment of MASP2. MASP2 interacts with this stable base and then recruits in MASP1. MASP2 autocleaves, cuts itself, releases that, releases one portion, MASP2, autocleaved, binds, recruits in MASP1, which will also autocleave and bind. The MASP1, MASP2 cleaved bound to MBL or phycolins will then recruit in C4 and C2. What we see happens is C4 is recruited in, C4 gets cleaved, A and B portion. A goes off and acts as a attractant for the innate immune cells. C4b stays and then helps with the cleaving of C2a. C2 cleaves A and B. B goes away, acts as an attractant. C2a stays. So what you end up with is MBL hexamer with MASP1, MASP2 bound, or phycolin hexamer with MASP1, MASP2. And then you also have bound to these, because these now act as a stable recruiting platform. You have C4b, C2a. C4b, C2a complex together to form C3 convertase. C3 convertase takes floating by C3 that has been recruited in, cuts it. C3b binds to the surface. C3a initiates inflammation, the red swelling around areas of infection. So, MBL, as well as the phycolin, recruits in, once it's stable, recruits in MASP1, MASP2. MASP1 cleaves 2, or 2 cleaves 1, depending on who you're reading. But MASP1, MASP2 must be there and activated to cleave C4 as well as C2. C4 stable to MASP1, MASP2 will then allow for C2 to be cleaved. C4b, C2a acts as C3 convertase. So when you look at it here, you can see that, well, here the blue hexamer, that's either going to be MBL or Ficalin, doesn't matter. Brings in MASP1 to MASP2, all of that's stable. C4 gets cleaved. C4 cleaves C2. B goes off, becomes an attractant. C2A binds to C4B on the surface. This cuts, this acts as C3 convertase to cut C3 into A. The inflammatory inducer in C3b becomes deposited on the surface. Are proteases. They act just like MASP1, MASP2 did, and they cut C4 and C2. You see here, this is an transmitting electron micrograph picture of, I forget what the magnification is, how many hundreds of thousands it is. Oh, it's 500,000. Shows you the structure of C1. So here is the individual trimers, and 1, 2, 3, 4, 5, 6. So the trimers can either bind directly to the constant, the FC region of antibodies that are opsonizing a bacteria, or they can bind to some sugar glycoproteins on the surface. However they bind to it and they're stable, C1, C1s, C1r will begin cleaving 4, 4 cleaves 2, and before you know it, you have a cleaved C3 bound to the surface. So when you look at the classical pathway, C1, C2, C4, C3, each of the different subunits is either acting as a mediator or directly binds to bring about the activation of C3b. When you look at the different parts, proteases, C1 keeps everything together, acts as a stable platform for C1r, C1s to activate C4 and C2. C4 binds, gives a place for C2 to be recruited to. C4a, mediator of inflammation, weak. C2a binds to C4b, acts as a C3 convertase, precursor of vasoactive C2 kinin. Means it's going to help the capillaries to dilate, allow more fluid through. C3a, peptide mediator. This is going to be intermediate activity, a stronger mediator. It's going to kick off the true inflammation at the site of infection. So this is the one they think may predate mammals, may predate most animals. I'm sorry, the early animals may have had this one. It has been known right now, as you're listening to me, somewhere in your body this pathway has just kicked off. Whether you have an infection or not, this one is a hair trigger. What happens is C3 just spontaneously breaks at that disulfide bond. B goes down. C3b recruits to its binding site, factor B. Factor B gets cleaved. You get factor D comes in. BA is released. BB stuck there with C3b and factor D. And once this starts, it just spontaneously causes other C3 to break. Now, this is a spontaneous, like I said, a trigger response. It's a hair trigger response. So we have ways to either keep it going or to make it short-lived. C3b just spontaneously deposits, then you get BB in, factor D and all that. Well, it falls apart very quickly. Falls apart unless it is stabilized by factor P, properdin. Turns out on our cells, factor P, the properdin, will not adhere, will not stabilize. So the C3 converts all on its own, it deposits, and then it falls apart within seconds. So there was never a cascade to really start. On the surface of a bacterial cell, factor P can bind, stabilize, and then you get the kickoff of the cascade.